And we're live from the Mandalay. Yeah, nobody watches UFC. Okay. So I'm back. I look the same, as a matter of fact, um, which would be no surprise if I was in the woods because I'd be in the same clothing without taking a shower for 8 days. So I will shower before I return. I'll make that promise to you guys. That way you don't have to smell my disgusting self. But obviously, today is the same day, right? We are here on Thursday. I am here, and you right now are actually all the way down there listening to a presentation in the cafeteria. I hope that it's going grandioso and that you're not bored and that you've learned some things and that Bella has taken a liking to their program for welding, and that's what she wants to do for a living. Be a fantastic job. I have a good friend of mine that welds on a pipeline and makes like 130 grand a year and gets 6 weeks of vacation. So it's not to say that you need to go to school for 44 years to make good money in this world. Don't have to. So for those of you who aren't into that, and I know I'm kind of talking to the cream of the crop for the sense that, y'all are only 14 of my senior class, and so most of you do plan to go on to higher education. That's fine. I'm not telling you not to do that. Not telling Claire not to go into dentistry, even though it was not for me. I was very good at what I did with dentistry, but did not enjoy it. For Claire, it may be the thing that she loves. I just want Claire to discover that early as she is going through that process, and that way, if it's not for her, she doesn't have an education that's useless. My education is not useless. It's helped me greatly. But what I mean is that she has put her effort into something that she intends to do long term. Okay? That's the same way for every one of you. Okay? For Tyler, we all know that nothing's really going to help him. But for everybody else, we know what we got going on. Okay? So let's give that our best effort. Hopefully, this week's been great for you guys. I really, really, really do want to say a big thumbs up and I'm thinking about you, I'm praying for you, and I hope all goes well with the cheer and dance competitions. All of our all-stars and whatever else is going on, I hope it goes fantastic. I want you to understand we don't always get what we want, right? So what I mean is just because you do something, and just because you fight hard for something does not mean that the result is always what you want. I can't tell you that I fought hard. I can tell you that I felt like I tried as much as my body would allow me to in our faculty basketball game. I had shots that I could not have let go out of my hand any easier at all. That it did exactly what I wanted it to do. And those shots went in the rim and touched the net and came back out of them. We lost a game. It's not fun. I didn't really enjoy losing. But the reality is that even though I wanted something, I didn't get that something. I would have preferred to have put 10 3-pointers in the hole. I think I shot all of them up during my practice round. I would have preferred to have done that, and I'd have been fine. But it's not what happened. And so I don't want you to hinge your life on the outcome of something like that. I don't want Emery to spend all of her time thinking to herself that if she makes All-Stars, that somehow that is now going to make her life what it needs to be. Two things happen when you do that. Number one, we get let down, and we get let down frequently. We don't win all the time. And number two, and I think the most important about this, is that when we put our livelihood, our stock, our this is everything to me into it, and it does fail us, we don't know how to overcome that. So I'd like for y'all to think about things in your life where you have done that. For some of you, this is not me being ugly, but for some of you, you have put all of your stock, all of your livelihood in parents always being there for you. And for some in this room, I'm sure your parents weren't. Not because they didn't love you, but because your parents separated, your parents divorced, your parents were going through their own struggles. And during that time, you felt like you were left out. And because you put all of your stock in that individual or those people, and they weren't there for you, it crushed you. Because you put all of your stock in getting a college scholarship to some certain school, or to play basketball or softball or whatever else it was, and it didn't happen, you can't overcome it, because the one thing that you were focused on is the thing that didn't succeed. I have a choice. I'm, again, 2 days away, and as you're watching this now, I'm there or on my way home or something. I'm going to go hunt an elk that has a 10% success rate. There's a 90% chance that I'm going to come home and tell you that either we froze to death and had to come home, that we got sick because of altitude sickness and couldn't make it, that somebody pulled a muscle and we had to get hella padded out of there or something. There is a 90% chance that we come home unsuccessful, and only a 10% chance that we do come home successful. I get to choose to put all of my stock in this trip is only successful if I shoot an elk and harvest him and take him home. Or I got to be smarter than that and say, I got to put my stock, my focus on something else. And all of you know, all of you, there cannot be in 100 gajillion years anything that you and I should put that type of focus and effort in than in God himself. There should be nothing else. There should be nothing else in our life that we think about on a constant basis to the point that we put all our eggs in that basket, knowing full well that if the basket were to turn over, we lose them all, than God. Because he does not fail. He does not make mistakes. And he will never leave you. There's 3. Ever. So, as we go to go cheer or dance, I'm praying for y'all. I hope you do awesome. I really do. But don't let it define you. If you win, don't let it define you. If you lose, don't let it define you. You make living as a young Christian man or woman, you make living as an example to other people, no matter what they do with their life, no matter what they do on Friday and Saturday nights or whatever else, you make living the way you're supposed to the only thing that matters. If you make that your focus, then I assure you, you'll get through so many other things. If you make other things your focus, if you make the boyfriend you're dating right now your focus, and then, like most boyfriends, he chooses to do something stupid, and it crushes you, then the reality is that then how do you overcome that? Well, you don't get to. You don't get to overcome that because then you thought, well, you know what? Everybody else is going to be this way. The next one's going to be this way, whatever it is. And the reason why you feel that way is because of you're putting everything on him, of saying, this is my all. It can't be. I don't want biology to be your all. I don't. I instead want you to be willing to focus on the one thing that matters. I assure you, if he does matter, the other things will still come in. You know, when he talks about the ideas of a man shall not eat, right, shall not work, neither shall he eat. If he talks about the ideas of God is wanting us not to be like the sluggard, he instead tells us to consider the ant and to be like the ant who works nonstop every day. So when we put God first, it doesn't mean you're going to ignore my biology. It means you're going to put more focus into the biology. And the reason why you did is because he says don't take things easily. He says work hard for what you need to get. That's what he's telling you. And so you would do that for biology. You would do that for your relationships with people. You would do that for your boyfriend and girlfriend relationships, your friend relationships. You would do that for teachers. You do that for parents. You do that for siblings. So I know I'm on a soapbox this morning, afternoon, whatever it is for you guys. But I hope you do awesome. But I want you to know that even when you fail a test of mine, it has no bearing on who you are. It may have bearing on what you did. Did you study? Did you spend time? Did you give the effort you should have? But it doesn't have bearing, or shouldn't have bearing, on who you are as a person for the long haul. If you allow that to happen, it's because you're focusing on the wrong thing. So, good luck in the competition. Hope you do awesome. Can't wait to come back and talk about it, how it went. Hopefully everybody did fantastic. And let's now get into the joys of active transport. Remember, passive transport, no energy required. Active transport, yep, we're going to spend some energy. Most of the time, it's because we need to get things out of a cell. Most of the time, it's because we need something in the cell. If we're bringing something in the cell through active transport, there is about a 900% chance that we're only bringing it in because we have to have it, or it's going to produce more energy than what it costs us to bring it in. Okay? If it's going out of the cell, at times we may expend energy that we never get back. That's not always the case. When we think about sodium, potassium pumps, for instance, we get that energy back. But at times, we are going to expend energy because we need to get something out of the cell because it's detriment to us, right? Some form of waste, and we had to express it outside the cell through something perhaps outside of, let's say, exocytoses, simple exocytoses that didn't require any energy. So let's think again. Active requires energy, passive, no energy. Okay, just keep that in our heads. I'm going to move me around again. Maybe I'll just talk like this and y'all keep up with it. Maybe not. No one got that joke. It wasn't really a joke. All right. Active transport. A cell must expend energy to move a solute. Look at this. Look at this. We already talked about this yesterday. Against the concentration gradient. So if I say against, what do you know? Areas of what? Are we going high to low again? Nope. We're going low to high. Energy molecule ATP supplies the energy for the most active transport. And we're going to look at 4 stages. There's only going to be 3 on your notes. I'm going to show you what the 4th one is because you're going to have to know them for my test. And we'll go over that in just a second, okay? Look at our checkpoint question. Cells actively transport calcium out of the cell. So that means it's inside, right? So they transport it out of the cell. It says, is calcium more concentrated inside or outside the cell? Which one is it? Outside. Why? We're moving it against a concentration gradient if it's active. We're going from what? Low concentrations to high concentrations, which is against. Down a concentration gradient is high to low. Okay? Any questions? We're all okay. Yes, no, possibly, maybe. Okay. Here we go. So this is going to be the first step. You need to know these in order, and you need to be able to explain them. All right? So we've got a transport protein, and we have a solute. The solute has to bind to the protein first. So in other words, it basically stays open. In this case, it's going to stay open until we have 2 of these bind. It will not close until it's got both of them in its spot. You can see that clearly drawn for you here as we're waiting on this solute to get here. This one's here. It is not opened. This is not closed. Okay? So solute's going to bind number 1. ATP is going to provide energy for the change of this. Okay? Step 2. So ATP is going to close this gate to open this gate. Everybody's got that? That's our second step. This is technically 4th step as it closes back. I want you to put 3 in between here, which is the solute leaving to the outside of the cell. Okay? Technically, they have this together in this case. I want you to separate them. There are 4 steps you're going to give me for the active transport of things. Solute binds to the transport protein. ATP changes the shape of the protein. Thirdly, the solute leaves the actual transport protein to the outside of the cell or inside, depending upon which way we're doing, because we can move it in and out, okay? In this case, we're moving it from inside outside, okay? And then lastly, we're going to close this back. Once the solute's gone, its door wants to shut back. It's kind of like a self-closing door, okay? When there's no solute present and ATP has been expended to have changed the shape, that would be us using energy to open the door. When we let go, the door closes itself down, okay? Should be Pretty simplistic to get you on that one, okay? All right, so everybody's good? 4 steps. Let's make sure that we got that. Put that in your head. We'll go over that on our review so that we're all on the same page. Okay? Solute binds, ATP is going to create the energy. Solute leaves, protein is going to return to its shape. Okay? So the more solute you bind, it'll continue to do that process. Active transport. Why? Because of this thingy right here. We had to spend some energy. Okay? All right. Show you here our sodium potassium pump, right? This is our 3 for 2. I'll show you how this works.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
So, sodium-potassium pump, okay? Very important pump in the regulation of all sorts of stuff inside of a cell. It is a 3 for 2. You're going to have to memorize this. 3 sodiums out, 2 potassiums in. She couldn't have explained it any easier. Nothing will happen, right, until the 3 sodiums are there and the actual phosphate group from the ATP bonds right here. When it does, it changes shape. As it does, the sodium leaves. Potassium then can come in. Same thing happens. It will change its shape again because the potassium's there. The phosphate group can be taken off. Off we go to repeat the same process. Okay? This is called active transport. Why? Did we have to use energy? Yeah, we had to get the phosphate group from an ATP. That's where it came from. Okay, as we expended it there. So just keep that in mind. Again, look, we're against concentration gradient. Why? Well, because we just moved, lastly, watch this, on the end, we moved 3 sodiums here against all of these. That's from low to high. That ain't from high to low. That's from low to high. So we're against the concentration gradient. Okay, and again, you can notice all these positives right here, sodiums and potassiums, right? Part of what we're doing is that we're changing this, what we call membrane potential, okay, here. Because of that, in other words, there's a change of charge from the inside to the outside because, look, we're putting 3 positives out and only bringing 2 positives in. But also, these positives don't want to go through this membrane, right? What do we say? Polar things and things with a charge. So because of that, what do we have to do? We've got to turn around and help it. We do so through a protein, transport protein, and actively, okay? All right. 2 mechanisms to move large molecules across membranes. These are large things. This is not potassium, okay? This is big bulk transport, okay? So this is the ideas of exo and endocytoses. Exocytoses, the leaving of things. Endocytoses, the bringing in of things, okay? Endocytoses has a few different types, right? Phagocytosis, pinocytosis, let's see, receptor-mediated endocytosis. So a few different types. Exocytoses basically has receptor-mediated exocytoses and then pretty much nothing more than just exocytoses, which is the process of a vesicle going to the edge of the membrane and then it bonding with the membrane and all of the junk that was inside of it going to the outside to the ECM or the extracellular matrix. Okay? So can't do anything besides memorize exo and endocytosis. No need to complicate the situation. It should be pretty straightforward and easy. Just put those things in your memory bank and be perfectly fine. Again, you got to think vesicles. Vesicles play a huge role in this. And you got to think that I'm going to ask you on this test, which one of these 2 things causes a cell to grow? I'll get into that here in a second with you, okay? All right, so on the endocytoses, I told you there's 3, and there are technically 3. For your information, I don't mind you giving me these 2. So phagocytoses is the bringing in of food vacuoles, okay? This is not the bringing in of liquids. It is the bringing in of food. Phagocytosis is what we would typically refer to as the feeding process of a cell. So it brings food particles in. Receptor-mediated endocytosis, basically we're going to bring in solutes, specific solutes that bind to receptors, and then the membrane will close around it and bring that into the cell because we need those things in. So another way to bring things in that case, right? In other words, we couldn't get them to come in through just simple diffusion, and instead we did so through pretty much like swallowing them, reaching out with our fingers and bringing those things in, right? So phagocytosis, put that with you with the ideas of grabbing around food particles primarily, right? Receptor-mediated endocytosis is around solutes, and put together in your mind the ideas of vesicles. We'll see some pictures here in a second to help explain this a little better. But it's just going to do nothing but bring vesicles. Those vesicles then can come in. Lysosomes can eat them, especially with the phagocytosis, or what we call a phagosome, which would be nothing more than a vesicle of food. That's all that it is. The lysosome would come, join with it. The lysosome would put off all of its enzymes to help digest it. Hydrolysis occurs, breaks it down. We'll use those parts for whatever we need to do for the cell. So notice this question at the bottom. As a cell grows, its plasma membrane expands. Does this involve endo or exocytosis? The answer is what? It's always exo. And there's a reason. As things are brought to the outside of the cell, the vesicle that is there adds itself to that membrane. More phospholipids. As we bring things inside the cell, we basically use the membrane as a sac to pinch off and to grab things, and so we lose parts of the membrane on the outside. Okay? That process is constant, so that's why the cell doesn't just shrink because of endocytosis, because we also have exo going on at the same time. All right? Golgi apparatus, remember, sending all those things. So what I want us to get a concept of is the idea of which one of these two things is making the membrane grow or shrink. Okay? And as we look at some videos, you'll see this. All right, so this is, right, a food particle. You can see these pseudopods, right? Pseudopodium is what we call this, but pseudopods, meaning false arms. They are going to grab around this. You can see a receptor-mediated, so in this case, we have some kind of solute out here, and they bind specifically right here. You can see how they've joined. What happens is, in this case, until they bind, this won't do this. Okay? And so what will happen is that once it notices that this has occurred, then it will start sending these pseudopods around so that it closes. So this and this will join, pinching off and creating this, and this is still going to be together. And again, since this is endocytosis, the bringing in of things, very simply, we understand that we're going to lose some phospholipids to the cellular membrane, and the cell's technically going to shrink, technically, okay? Though it's temporary because we're going to fix that process. Okay, same picture, just larger for you to see. That's us bringing in food. Of course, a lysosome is going to attack this food vacuole so that it can dissolve, harvest basically the food into energy for the cell. Same thing here with our receptor mediated. Okay, so I'll show you this video.
Endocytosis is the movement of materials into a cell via membranous vesicles. Exocytosis is the movement of materials out of a cell via membranous vesicles. These processes allow patches of membrane to flow from compartment to compartment, reminding us that a cell is a dynamic structure.
Part of why we call a membrane a fluid mosaic model, right? So again, endo, inside, exo, outside. Think exit, okay? That's all you got to think, right? Enter, exit, all right?
Exocytosis is the movement of intracellular vesicles to the plasma membrane, where they fuse with the membrane and release their contents into the surrounding fluid. This process occurs predominantly in secretory cells, such as mucus-producing cells or pancreatic cells, that secrete enzymes into the digestive tract.
So notice what type of cells primarily go through the process of exocytosis. It's not in your notes, so you better write it down somewhere. Secretory, right? Right? Secretions. That's the word that we mean by that, okay? So they're putting things off. If they're putting things off, there's our exocytosis. There's our exit, okay? Should have a video here of our Pinot, right? I told you this is your third one, okay? This is the bringing in of fluid materials. We'll see this here.
In pinocytosis, or cell drinking, the cell engulfs extracellular fluid, including molecules such as sugars and proteins. These materials enter the cell inside a vesicle. Epithelial cells and capillaries use pinocytosis to engulf the liquid portion of blood at the capillary surface. The resulting vesicles travel across the capillary cells and release their contents to surrounding tissues, while blood cells remain in the blood.
So again, pinocytosis, think water or cell drinking. It's bringing in liquids, not water, because it doesn't have to do this to do water. Remember, water can flow back and forth, but it's bringing in the liquids from the outside, okay? As obviously we've already talked about phagocytosis being the process of eating, and here you have an example of that here.
In endocytosis, membranes invaginate, or pinch in, to form a vesicle, moving the enclosed materials inside the cell. This process can take different forms, each involving its own specific cell machinery. In phagocytosis, or cell eating, the cell engulfs debris, bacteria, or other sizable objects. Phagocytosis occurs in specialized cells called phagocytes, which include macrophages, neutrophils, and other white blood cells. Invagination produces a vacuole, which usually fuses with one or more lysosomes containing hydrolytic enzymes. Materials in the vacuole are broken down by these enzymes and degraded.
I want you to get a concept, if you will, of the idea of how these things are being brought in and then what would happen to them. Not just the how they come in, what would happen. Look at what just got said. Why on earth would a cell bring bacteria from outside the cell into the cell? I mean, it's more than content, theoretically, to leave that junk outside so that it can't affect us and do anything to us. So why on earth would it turn around and bring it inside? That's right. What's inside of a cell that can help it deal with that bacteria? Well, see, if it keeps it in a membrane-bound sac, vesicle, if a lysosome then comes to give it its enzymes, then you instantaneously have what? You have the ability to digest, break down, and destroy that bacteria. Can't really do that out there in the extracellular fluid. So that's why sometimes we bring bad things in, because the cell then has the ability to function and deal with them. Okay? So again, phagocytosis is pretty much the feeding process. Primarily, we're bringing in solid things that are out in the ECM. Okay? Primarily. Sorry, my wife's texting me about her chicken missing. Yeah, go figure. All right, receptor mediated. Remember, in a receptor mediated, we're basically bringing solutes in, but we have receptors that are made to bind to those things to say, hey, now it's time to bring our pseudopods around and bring it in. Okay? That was an animation, I think. It was. I didn't play.
Unlike phagocytosis and pinocytosis, receptor-mediated endocytosis is very specific. It is triggered when membrane receptors bind to specific external molecules, such as protein cholesterol complexes or proteins bound to iron. Membrane vesicles pinch off, and the external protein and its cargo are brought into the cell.
So again, remember, these receptors exist all on the outside. By the way, let me ask this question. Anybody have a clue what this receptor is made out of? Emery, what's it made out of? It's not a phospholipid, right? It's a what? It's a protein. It can be an integral protein or peripheral protein that's nothing more than a receptor site. That's what it is. It's basically built into the membrane that originally came from ribosomes out of the nucleolus to the rough ER. The protein was then built, packaged, sent to the Golgi apparatus. The Golgi apparatus literally sent a vesicle to the cell membrane. It became part of the new cell membrane. End result is now you have a receptor to find and to locate, right, is what it's doing, these things. So shouldn't be some drastically crazy concept of how this got there. Most importantly, just understand all this is again, it's the bringing in of things. Specifically, we're trying to bring in some form of solute that we need inside the cell, and we do that through this case of receptor-mediated endocytosis. Okay. All right. Let's look at energy in the cell. This section is not going to be entertaining to some of you. Okay. There's nothing I can do about that whatsoever. Sorry, I keep getting off camera. Nope, wrong way. I think that's just because, yeah. There we go. Now I don't have to feel like I'm off camera as much. All right, so it's just going to be about really physics, and there's not much you can do besides get this kind of stuck in your head so that we don't miss this long term. All right, kinetic energy is the energy of motion. Write it down, know the definition, nothing you can do. Potential energy is energy stored in the location or structure of matter and includes chemical energy. So chemical energy is a form of potential energy. And then you got to know some laws of thermodynamics. You're going to have to list these on my test once we get there for review, etc. Energy can change, cannot be created or destroyed. And when energy transforms, they increase disorder or what we call entropy. And typically what will happen is that the energy is lost as heat. It doesn't mean it's gone. Yes, we lose it from what we're dealing with, but it is put off as heat. So, as cells function, right, those cells are going to go through a process which expends energy. If they expend energy, they cannot create energy from nothing. So in other words, energy can change form. It cannot be created or destroyed. So they can't create it from nothing. So the energy that already existed in glucose was just turned into a different form through ATP. That's what happens. And then that energy is going to change, and there's going to be entropy or a degrading of those things, a disorder. And what that means is that there's some energy that's going to be lost through actual heat. Hence why when you work out, your body is hotter. Why? It's because your cells are literally working. And because your cells are working, they are putting off heat, right? No different than a car, right? Same general concept. Okay. All right, here's an example, right? Gasoline and oxygen causes combustion of a vehicle, right? So in other words, the energy is created, right? The kinetic energy is the tires. Why? Because it's movement, right? Which is very much relying on the energy that existed through this original potential energy, okay? There's going to be heat given off, right? Entropy has caused a disorder, and so some heat is going to be given off. That's why your engine is hot when you touch it. And the end result is that we're going to have waste products, right, of carbon dioxide and water. Look at your cell. It is absolutely no different. I'll show you this in the next two slides because it'll be a little easier to see. Look at your cell. Glucose and oxygen, right, through cellular respiration, is going to give us ATP, energy that we need for cellular work, right? We are going to have heat energy put off, okay, because of entropy. And then lastly, we're going to have waste products, carbon dioxide and water. Just like a vehicle, okay? Just like a vehicle. Show you here. Kind of what
Energy is the capacity to cause change. Kinetic energy is the energy associated with motion. Energy that is not kinetic is called potential energy, and is energy that matter possesses because of its location or structure. Energy is neither created nor destroyed, but converted from one form to another. This property is called the conservation of energy. The bow and arrow start out with low potential energy. As the archer's arm pulls back the string, kinetic energy from the arm's motion is converted to potential energy in the tense bowstring and arrow. When the bowstring is released, the stored potential energy is converted into the kinetic energy of the moving arrow. When the arrow hits the target, its motion ceases. If energy is neither created nor destroyed, where did the energy of the flying arrow go? Let's rewind and look at kinetic energy in more detail. When energy is converted from potential energy to kinetic energy, Some of the energy can be used to do work, but some energy ends up as heat, a type of kinetic energy. Heat is the random motion of atoms and molecules. As the arrow flies through the air, heat is generated by friction between the arrow and air molecules. When the arrow strikes the target, all the energy becomes heat energy. The heat energy is rapidly transferred into the air and spreads out. Heat energy is a very disordered kind of energy. It has the highest amount of entropy, or disorder, of any kind of energy. Where do our muscles get energy to perform work, such as pulling back a bowstring? Our bodies use the chemical energy from food to perform work. Chemical energy is a form of potential energy. When your body breaks down food molecules, the stored potential energy from food can be converted to kinetic energy. The stored chemical energy in food is released in your muscle cells during the process of cellular respiration. Using oxygen, cellular respiration converts chemical energy from food to another form of chemical energy called ATP. Water, H2O, and carbon dioxide, CO2, are byproducts of cellular respiration. The potential energy of ATP can be converted to kinetic energy. Some of the kinetic energy is used to do useful work, such as pulling back a bowstring, but the heat energy that is generated cannot be used to do work.
Okay. I cannot say this enough because I'm going to have this entire PowerPoint on RenWeb and available for y'all. This 100% is the thing you need to get a concept of. You probably should watch this video several times if you're missing any ideas behind it. Remember, kinetic energy is movement, and that includes movement that is heat, atoms moving. Okay? Potential energy is the ability for it to get to that point, to actually do work, to have heat energy or whatever else it is. So we're going to have several questions on our test about chemical energy and potential energy, and this video is a key one, in my opinion, to gain some concepts. I'm not going to play it again for you in this class just because of time, but you can watch this over and over if you need to so that you get this concept, okay? So again, put it all together. Put how they're related to each other. Put how potential energy can create kinetic energy, okay? Those things you've got to try to gain a general concept of, okay? All right, exergonic reactions. Release energy, endergonic require energy. Typically speaking, they're going to suck energy in, if you want to think of it that way, okay? Metabolism encompasses all of a cell's chemical reactions, okay? All. Metabolism, the balance of everything in a cell. That's what that means. We're going to talk specifically here in just 1 second about how we control that metabolism. So, exergonic reactions release, endergonic require energy. They yield products rich in potential energy. In other words, they have brought in energy and now they have something that is ready to go out and do, okay? So, endergonic leads to exergonic, if you think of it that way, okay? Trying to give you time to write this down. Not being weird. I'm just staring at you. Just kidding. All right, so here is the amount of energy that is released from where we started, right, our reactants, to our products. The amount of energy released. Take a look right here, okay? This is the energy that's put off, all right, as we're trying to go from reactants to products. Amount of energy released, okay? All right, I'm going to show you what that means as we look at the ideas of enzymes here, okay? So this is the potential energy of molecules, all right? Potential energy, meaning it has the ability to. So we are, in this case, we are endergonically bringing energy in. Here's our reactants. We have stored energy to create a product that has got what? Look at this. The amount of energy required, right? Okay, that's the change from here to here is what that's showing you. Okay? These products basically, okay, now have far greater potential energy. Right? They can do more. Why? Because this has been put with it. Okay? We've added this energy to it. All right? So, ATP, right? Adenosine triphosphate powers basically everything that happens in a cell. What do we do? You're going to have to know this for your test. What do we do when we go through this process? We have got to literally remove a phosphate group off of ATP. Okay? If you notice this, it says that this is involved in chemical, transport, and mechanical work. It is. Okay? There's a chemical change. There's a transport of the phosphate group. There's mechanical work in doing so. All these things because we're taking the phosphate off of the ATP and creating energy because the phosphate is bonded to it in such a way that it does not really want to let go. So the breaking of that bond releases the energy that's there. Okay? That's what that does. All right? And again, look, we can go exergonic to endergonic in ATP. We can give off a phosphate group and then turn right around and have that phosphate group be put right back to us. So we can be exergonic in the giving off of energy, and then we can be endergonic as that phosphate group is brought back to us, and as we have it as part of our ATP again. So we can go from ATP to ADP, right? Adenosine diphosphate has nowhere near the energy that an ATP does because we can't break that last phosphate group off. And then we can put the phosphate group back on. And as we put a phosphate group back on, we've done what? We have given it an endergonic reaction. We've added energy to it and increased its potential energy for the future is what we've done. All right. So again, I'm aware this is physics. Some of you have not even taken it or aren't going to. I don't know. You've got to get some concepts behind the ideas of bringing energy in and then what can we do with that energy. Okay. There's your ATP, right? Take a look. Why would we bring water into here? What's this process called, Kate, again, when water comes in? Nice, Kate, hydrolysis, right? Why? Because, well, guess what? We stuck, right, a hydrogen here and an OH here, right? And then we turned around and we broke this bond, so we have a phosphate floating around and we have energy. Can we then turn around and put this phosphate group back on here? We can, okay? We can bond the phosphate group back and we can also then break it off again, right? Adenosine triphosphate and diphosphate, okay? Notice these three things. Here is the chemical work that's involved, right? This is very simply, right, this phosphate group being taken off, okay? Instead, we've done what? We've added the hydrogen here, okay? We have transport work. Why? Well, because we needed to use this phosphate, okay, right here to help to do what? Remember, we talked about this, did we not? Our active transport by bringing in and out things in and out of the cell. So you can see them here, right? The phosphate was involved in transport work. Can the phosphate right here be involved, right, in doing something else? If you take a look, I'll try to get myself out of the way. Here you go, right? Look, every time, look what we have left. ADP plus P. Why? So we can repeat this process, right? Because we need to get back to it and do it again. So here we are, right? This is a motor protein, is what this is. You can see it, okay? Right here primarily. We can do what? We actually can shift, we move its location, okay, from where it's at to where it's at. Why? We do so with the phosphate group. So in other words, ATP is involved in basically every form of work that ever happens ever inside of a cell. If it's chemical, if it's transport, if it's mechanical, doesn't matter. ATP is involved in it, okay? And guess what? It's a renewable source, okay? It's like what we define as solar energy if you think of it that way, okay? It's not really solar. I'm saying it's renewable because we got to have it. We got to do it again, okay? This is your loop, right? Okay, energy comes in from cellular respiration, right? ATP synthesis requires energy. In other words, the making of ATP requires energy. Now we have ATP. We're going to release that energy by breaking ATP down, right? Hydrolysis. Energy is going to be put off for the cellular work that's going to happen. We then have to turn right around and we've got to rebuild our ATP so we can come this way through cellular respiration to do this process over and over again. So it's an exergonic, endergonic, exergonic, endergonic, exergonic, endergonic. It happens over and over and over and over and over and over again, okay? And we got to, because we got to continue to have energy for all the things that happen. There's no letting up. We don't have a choice, okay? All right, enzymes. Enzymes are catalysts. What does that mean? They technically don't speed up a reaction, even though that's what they're doing. They instead do something. They decrease the activation energy required for the reaction. What it took to get it kick-started, they fixed the problem, okay? If it was 4 steps above us, what they literally did was they said, how can I make 4 steps become 1? It's easier to overcome that 1 step than it is to come over 4 steps. Okay, so that's how they speed this process up. Enzymes, what are they? They're proteins, everybody. How do we denature proteins? That's exactly right. Temperature, right? And pH. We talked about that 2 tests ago. Temperature and pH. You're going to have that issue pop back up here later on, okay? Yep, almost died there. All right, all this you'll see elsewhere. It's not your bell, by the way. Not unless this video got played 9 minutes into class starting. So watch this. This is our energy required to break right here. That's what it takes. It's a hump. Look at it. So an enzyme basically comes in and it lowers the activation barrier. Okay? So it reduces the activation energy required is what it does. Therefore, it takes less energy to be expended to break this bond. Same thing could happen in making this bond. That's what it's doing, okay? It's a good example. Here again, large activation barrier, okay? Small activation barrier. Same general concept, okay? You could see all of this, right? Reactants, move this out of the way. Reactants and products are what we started with, right? This red represents what? It represents what's happening for the activation energy, the hump required to get started with an enzyme versus this huge hump that's required here without the enzyme. Okay? Look, same products. We got to the same place. Okay? Exact same place. But we did so mucho easier here. All right? Mucho easier. And this is, of course, look, we have this energy, right? Here to here, the change of energy from reactants to products. All right? Enzyme catalyzed reaction, right? Enzyme catalyzed reaction, red one. Okay?
If exergonic reactions occur spontaneously, what keeps molecules from breaking apart and cell chemistry from racing out of control? For any reaction to occur, even a downhill reaction, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, E sub A. This required energy input represents a barrier that prevents even energy-releasing exergonic reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules, referred to as the enzyme substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction.
Okay. It appears as if I'm at like 47 minutes. So for today, I'm going to stop. I don't have much left to go, and I will go over that information on our review day because it's nothing more than basically a continuation of this idea of enzymes that he just talked about, okay? So I will have one more video on Thursday, which will be your review day. I will tell you everything that you've got to know basically on the test, and you'll be ready for that, okay? So hopefully I'm not still talking and you're already out of the room because the bell's gone off because I guess it is what it is, okay? Hopefully we learned a lot today. Look forward to seeing you. God bless. And again, do well tomorrow at Cheer and Dance.